Tonight we're hosting Professor Sir David Baulcombe. He is the Regius Professor of Botany and Plant Sciences at the University of Cambridge. So please give me a warm welcome and thanking him for coming out tonight.
Oh, well, thank you very much, and thanks for the chance to come and talk to you this evening. As you can see, we have a little audio or visual problem, so there's going to be a purple background to everything that I say this evening, and I hope that won't detract from the ultimate message that I'm trying to get across. So there are several components to what I would like to talk to you about this evening. The first is in biology, and it's about the tree of life. So I work with plants, and so plants are sometimes seen as a little sort of niche backwater of biology as a whole, but that's not how I see it. Well, I would say that, wouldn't I, as Professor of Botany, but that they are actually quite important for food and fuel and all of that sort of stuff. But just in terms of understanding basic biology, I think Darwin summed it up quite nicely, which is that there's a great big tree of life and it's interconnected at the root. And so things that you find out about plants are sometimes relevant in the broader context, in the biomedical sphere, for example, and so on and so forth. So number one thing that I want to talk about then is about the unity in different living systems. I want to tell you a little bit about how to make some progress or discoveries in science, at least using some lessons from my particular experience. And then thirdly, I'm going to tell you about things that you find a bit interesting, actually, in the world of science itself. So the project I'm going to talk to you about this evening is one that started off several years ago when we were exploring in my lab a concept, very simple concept. It's a sort of immunization concept, if you like, called parasite-derived resistance. The idea is if you want to make an organism resistant to a parasite, then what you do is take a gene out of the parasite and you put it into the host organism. And the idea is that that will be a sort of molecular immunization process, and it will confer resistance against the parasite in the host. And this was a concept that was first reduced to practice with a bacteriophage, a virus that infects bacteria and their host bacteria. And we wanted to explore whether or not it would work in plants. So the idea is that when you've got a parasite and a host, in order for the parasitism to work, resulting in disease, that there has to be an interaction between a component of the parasite and a component of the host. And the thinking is, if then you introduced as a transgene a component of the parasite into the host, but one that is maybe not quite right, so it might lack a part of it, so it's actually not functional as a parasite component, as a pathogenicity determinant, but it still binds to the host component. What will happen in that scenario is when the host organism is infected with the parasite, it's got nowhere to go, so you don't get disease. Very simple concept, very simple idea, works with bacteriophage. Would it work with plants? That was the idea. So we were doing genetic engineering of plants. I know it's a controversial subject. It's what we use as a research tool. We could have a discussion about whether or not it's useful in agriculture or the real world a little later on. But anyway, this is what we were doing. We use a soil bacterium as a vector for transferring genes into the DNA of the host organism. You simply infect cells of a plant with the soil bacterium, and then you regenerate whole plants that have integrated DNA from the soil bacterium. So we can use that as a genetic engineering strategy by taking other genes that don't actually belong in the soil bacterium and introducing them into the soil bacterium itself. And so what we were doing, following this parasite-derived resistance idea, was taking genes out of a virus and using the bacterium as a shuttle, introducing them into a host. So the virus, we were using a rod-shaped virus known prosaically as potato virus X. I'm afraid plant virologists are not very imaginative. We could have done the experiments with potato virus Y, or potato virus M, or potato virus A, Get the idea? Anyway, they're all different viruses, and many of them cause diseases not only of potato but other plants as well. So we thought it would be useful to see if we could make resistance against this virus. So that's what we did, and we got resistance. We got amazing resistance. We got plants that were so resistant against potato virus X that you could take preparations of virus that were so thick they were viscous, they were cloudy, and you could rub them onto the leaves of the plants and they would not get infected. But there was something a bit wrong. And so what was wrong was, so what happens whenever you make transgenic plants, whenever you do anything in science, it seems to me, you get variation in your results. And so when you make transgenic plants, GM plants, some of the plants that you get out of the whole exercise are expressing the gene that you put in, and some of the ones aren't. So that's normal. But what was pretty weird was that the ones that were resistant, the ones that did not exhibit this mosaic symptom when we inoculated them, the ones that were resistant were apparently not expressing the transgene at all. Transgene was switched off. It was silent. We had other lines where the transgene was being expressed at a high level, and they were fully susceptible. So this is a bit weird, so you normally think that some of something is good and more of it is better. And here we had the exact opposite. So some of transgene expression is apparently no good, and none of it is absolutely brilliant. And then the intermediate situation is about halfway between the two. So that was all a bit weird. And we thought about it for a bit. And then what I realized was at the time, so there were other people doing experiments with transgenic plants at the time, and they were also getting strange results. So here's an example from some colleagues in California, and they were fiddling around with flower color. And what they were trying to do was to make flowers of petunias that were more darkly pigmented than are the natural ones. These look artificially black. They're sort of bluish if you look at them without the purple background. What they were doing was taking a gene out of the pigmentation pathway, putting them into plants in the expectation that the plants would have more darkly pigmented flowers. What did they get? They got this. They got some plants that were exactly the same, and some plants that had lost the pigmentation totally. And they also got these, which they called Cossack dancer, for reasons you can probably appreciate when you look at the shape of the pigmentation. So what we realized was that actually these phenomena were actually conceptually the same. So what we thought you could do is you could explain the flower pigmentation result if we had got the transgene and we had introduced it into a plant. And what we thought was probably happening was that the transgene was doing something that we ultimately called RNA silencing. And that this RNA silencing would be silencing not only the gene expression from the transgene, but it would also be silencing the endogenous flower pigmentation pathway. So that is why when you put a transgene into the plant, you got no pigment or loss of pigment. And in the same way, when we had our viral plants, so plants carrying the viral transgene, what we inferred was happening was that we got this RNA silencing business that was going on, and that this RNA silencing business, whatever it is, was not only blocking the viral RNA of inoculated viruses, the same mechanism was blocking the viral transgene RNA. So, the same sort of process could explain both sets of results, and it could explain the apparently anomalous result, which was that low transgene expression was giving us the high level resistance. So here's the little lesson, if you like, which is that when you're doing research, from time to time you will encounter anomalous results. Leap on those anomalous results because they are a little indicator that there's something going on that just doesn't fit with the paradigm. Thomas Kuhn wrote about all of this sort of stuff, normal science, paradigm shifts and all that sort of stuff. So when you get these anomalous results, make sure they're anomalous. We looked at our plants till we were blue in the face just to make sure that the transgenes were indeed off. But once you're sure that you've got an anomalous result, that is something to latch onto. And that's what we did. So we'd got this anomalous result then, and we'd got indirect evidence for a process that we called RNA silencing. We called it RNA silencing because the viruses that we were working with at the time were viruses that did not have a DNA phase in their replication cycle. They lived entirely in the RNA world. They didn't go into DNA. So we knew that whatever was happening was not acting at the DNA level. I'll come on to this a bit later on. It was acting at the RNA level. So that was, we could infer the existence of this protein, this process, but we didn't know anything about it. One of the things, though, that we could latch onto in our exploration of the mechanism was the specificity. So what we knew in the Petunia experiments was that the only gene that was silenced was the pigmentation gene. So it happened to be encoding an enzyme called chalcone synthase. But the only gene that was silenced was chalcone synthase, nothing else. In the virus experiments, the only virus to which the plants were resistant was potato virus X, not potato virus A or M or Y, or even some other strains of potato virus X. So what was going on here was very highly specific. And what we could infer was it was likely to be nucleotide sequence specific. So then nucleic acids, you all know about DNA, double-stranded DNA, A binds to T, G binds to C, and that sort of stuff. RNA can do the same sort of thing. A binds to U in RNA and G binds to C. But anyway, they can form base paired structures in exactly the same way. So when you've got sense RNA, the opposite strand, we call it antisense RNA. So what we could infer was that in this RNA silencing process, whatever it is, that there was most likely to be antisense RNA. So we could infer that the transgene was going into the plants, probably making antisense RNA, and the antisense RNA would then bind to the transgene, it would bind to the virus, and it would explain why both were blocked in this whole process. So that was fairly straightforward. The big challenge then was to find the antisense RNA. And actually, we spend a long time doing this. I had a very talented and able postdoc in my lab called Andrew Hamilton, a very persistent, tenacious young Scots guy, and he looked all over the place and he couldn't find this antisense RNA, but we knew it had to be there. And we finally realized we use a technique, gel electrophoresis, in order to look for these RNA molecules, charged molecules running through a gel, small molecules run faster. What we figured out eventually was the only place where these antisense RNAs could be would be in the bottom gel tank. They'd run through the gel and off the bottom. That had to be true, so Andrew adapted his technology and developed different methods for looking at these antisense RNAs, and eventually he found them, and they were these nice molecules here, about 20 odd bases long. They're short RNA molecules, and they're not just junk, they're very specifically sized RNA molecules. And when we were doing silencing of something, you had these short antisense RNAs corresponding to the target of silencing, and not when you weren't. So we were fairly confident that these antisense RNA molecules were at the heart of the process that we were sort of interested in. Now, at the time, in the rest of the tree of life world of biology, there was Andrew Fire and Craig Mello getting weird results when they were feeding RNA molecules to worms. You got people working on fungi, getting strange gene silencing phenomena. And people were doing things in mice and Drosophila and everything like that. A lot of people getting things where genes were being switched off, and it seemed as if we were looking at the same thing. And eventually it turned out that we were looking at the same thing. So Fire and Mello discovered double-stranded RNA as a key component of all of this. It turned out that our discovery of these little short RNA molecules was also common to both animals and plants when they were doing these various RNA silencing, gene silencing type of phenomena. So then we had a very nice period of a few years when it seemed as if every week you opened a journal and you could read a paper from Drosophila one week where they discovered something, and you could say, aha, that fits into the puzzle here. And then a mouse paper the next week would fit in there, and so on and so forth, and a worm paper the next week. And it all came together. And so then we got a pretty good understanding of how this process works. And it was so good that Nature made a cartoon that you can download from their website, or at least you used to be able to download it from their website. And what they've now done is to put on a much more structurally correct and actually much more complicated cartoon. And this is the old version that is actually a lot simpler, and it just fulfills the purpose that I want to this evening. So this is how RNA silencing works. And we're looking inside a cell here, so that lunar structure in the background is the nucleus of the cell. And for the sake of argument, this is an RNA molecule here, and it happens to be an RNA molecule, let's say, from the transgene in our virus experiments. So what is happening is that somehow the cell recognizes that RNA as being weird or foreign. And so it brings along an enzyme that copies the RNA from the transgene into a double-stranded molecule. And cells, they don't normally have double-stranded RNA. RNA is normally single-stranded, although it can be double-stranded. And there's an enzyme in cells called dicer, which dices these longer double-stranded molecules into the short molecules. And these are the short molecules that Andrew found in my lab. These short molecules initially exist in a double-stranded form, but then they bind to another protein, which we call slicer, dicer and slicer. Initially, it's in a double-stranded form, but then one of the two strands is lost. So then you've got slicer, and it's bound to a single-stranded short RNA. And this single-stranded short RNA is like a guide. So the slicer with its bound RNA roams around the cell, and it's looking for another RNA molecule with which it can pair using Watson-Crick base pairing. And eventually it finds it. So there you go, it finds it, it docks it, and slicer slices the targeted RNA. So then you've got an RNA which has been split into two halves, and cells have a little munchkin that comes along and it hoovers up these cleaved RNA molecules. So that's all quite nice. And so that is basically, it's how this process works. And it operates in plants and animals, and so it is part of the tree of life. So where does this get us? Well, one of the things you want to do, I'll come on to what this does in biology, but one of the things that you might want to think about is, well, what does this do? Is this useful? And I have pinned up in my office here this little quote here. This is from Robert Hooke, Micrographia, 17th century book of micrographs. It's an absolutely wonderful book. It's a book where he describes cells for the first time. He's looking at cork, and he describes the sort of structures that he sees in cork, and he compares them to the habitations of monks in monasteries, the monastic cells. So that's the first use of the word cells. It's the great prerogative of mankind above other creatures that we're not only able to behold the works of nature or barely to sustain our lives by them, but we also have the powers of considering, comparing, altering, assisting, and improving them to various uses. And this is the peculiar privilege of humankind. So when you do something, you want to do something that's useful. Or when you've found something out, you want to do something that's useful if you can. And it turns out that RNA silencing is actually extremely useful. One of the things that it's useful for is functional genomics. So you're probably aware that now, and I'll come on to this a bit in a moment, but due to a recent revolution in biology, it's now relatively easy to determine the complete DNA sequence of almost any organism. So then you've got this great big long string of A's, C's, G's, and T's, and you can infer that there's a bunch of genes in there. So then you wonder, well, what do these genes do? So not a trivial undertaking. But what you can do using RNA silencing is you can systematically go through the 30,000 or so genes that plants and animals have in them and knock them out. And then you say, what does this do to the organism? And when you know what knocking a gene out does, or what silencing a gene does, you can then infer what is the function of the gene. So you make collections of RNA or DNA molecules that result in RNA silencing being introduced into different cells, and you go from 1 to 30,000, depending on the organism and the number of genes it's got, and then you look at the phenotypes. And this is extremely useful. This is just one of the experiments that we did. We used a virus to introduce RNA molecules corresponding to, this was just a trial experiment we did, just illustrates the point though. We introduced RNA, these little small RNA molecules corresponding to a gene that is necessary for flower production. So what happens in the plant that has these small RNA molecules is it can't make proper flowers. And this is exactly the same as what happens if you have a mutation in this gene. But of course, when you make mutations, you do it randomly. What you can do is you can do this systematically and in a targeted way. It's been useful in plants. It continues to be useful in plants and in animals as well. So RNA silencing is useful in functional genomics. It's also useful as a disease strategy. So I showed you how at the beginning we were able to make plants that were resistant against potato virus X. Actually, that wasn't particularly useful. It was just a good experimental thing to do, nice experimental system. Potato virus X is actually known as the healthy potato virus because practically every potato that you buy off the shelves in your supermarket is infected with potato virus X. But there are other viruses that are a bit more of a problem. Here's one that is a problem for rice farmers, upland rice farmers in Africa. And they're troubled by a virus known as rice yellow mottle virus. And so what we did was to engineer rice varieties for them that were resistant against rice yellow mottle virus. So this is a wild type plant, and these are our various transgenic lines after infection with the virus. So again, we could have a little discussion later if you wanted to about whether or not using GM to do this sort of thing is a good or a bad thing. I think you can anticipate what my view is. You can also, or at least this is a little more hypothetical, although not totally hypothetical, think about using these small RNA molecules as a pharmaceutical, as an antiviral or anti-disease agent in animal cells. So the idea here is that you would synthesize RNA molecules that are targeted against a virus or against an endogenous gene that causes disease. And what you would do is in various ways you would couple these RNA molecules to fat globules or other compounds that would cause them to be taken up into cells. And the idea is that you can use these RNAs being taken up as a way of knocking out gene expression. And there's a bit delivery. So getting the RNA molecules to go into the cells is the big challenge here. But there are indications that this can be done. And I'm optimistic that it actually will be done eventually. And just think about it. If it works, it is absolutely fantastic because you don't have to do any of this horrible sort of structural stuff and working out which drug fits into which little pocket of the protein. If you've got a virus or something that you want to block, you just determine what its sequence is, and then you make an RNA that is the complement, and you put it into cells. So you've got your specificity built in this process. So if that works, it'll be absolutely fantastic. Anyway, these are just a couple of things you can do with RNA silencing if you want to, things that could be useful. Why do organisms have this whole process? What is its natural biological role? Well, one of the things we know is that it is something to do with virus resistance. So here's a nice little irony. So there we were beavering away trying to make plants that were resistant against virus diseases. But it actually turns out that this RNA silencing process is something that plants use naturally to protect themselves against viruses. So all we were trying to do was something that nature had been doing already. But that's only part of what it does. Just think about this. This is a process that existed in a primitive cell that is the common ancestor of plants and animals. It must have been there because it's present in both. So there's been a long time since the common ancestor of plants and animals. And so there's been time for diversification, and that's what we see. So the role of RNA silencing in animals in some ways overlaps with that of plants, but in other ways it's subtly different. And what has happened is, we think, is that this process, which probably, I mean, you can never know with evolution, but it probably evolved as some sort of defense mechanism against primitive viruses, has subsequently diversified to fill a number of different roles. And one of the things we've been able to do recently is to get a picture of the diversity of RNA silencing mechanisms in plants as a consequence of having access to new technology for sequencing nucleic acids, DNA and RNA. In my career, so I got my PhD in 1977 in biology, in molecular biology, there have been two revolutions. The first one was happening just at about the time when I got my PhD, and this was when gene cloning was just beginning. So it meant that we could look at individual genes as bits of DNA in cells. And so that then occupied the next 20 or 30 years of my career. And we could beaver away looking at individual genes and make careers out of it and all of that sort of stuff and learn a lot. But what has happened in the last several years is that we now are not only able to look at individual genes within a cell, we can look at whole genomes. We can look at all of the RNA molecules in a cell at any one time. We can look at the various ways that the DNA is packed into the chromatin of the cell, all of the DNA, using techniques that are based on what is known as next generation sequencing technology or high throughput sequencing technology. I'm not going to go into the details of it. I think for the purposes of this talk, all you need to know is a bit of kit that will fit, I don't know, just about this much, will determine the genome of a human being in a couple of days. And I mean, the power is just incredible of this technology. And we've used this to look at small RNA in cells. So having discovered these small RNAs, having found that they're made in transgenic plants, having found that they're made in virus-infected plants, what we wanted to do was to say, well, are they made just in regular plants? You know, plants that haven't been infected with a virus or had the insult of a transgene put into them. The answer is that they do. Plants are actually jam-packed full of these little molecules. This is a bit of data. I think I can just talk through it. The point is that in a plant genome, there are about 30,000 protein coding genes, as I've told you. There are also about the same number of regions within the genome which give rise to these little small RNA molecules. So you've got an enormous amount of sequence information that is actually represented as these little short RNA molecules. And this then gives rise to my title, the dark matter of the genetic universe. So what we think is that these little small RNA molecules in various ways, and I'll talk about this in a moment, have types of regulatory function. And what they're doing is a bit like, the hypothetical dark matter of the physical universe that's supposed to be stopping the universe expanding more rapidly than it is. We think that the dark matter of the biological of the cell, this RNA molecule, is somehow an integrator of regulatory pathways. And there are various lines of evidence for thinking about that. The point is there's loads of this stuff in cells. And again, this is sort of technical stuff. Within plants, there are lots of different pathways that give rise to these small RNAs, and they have several different roles within the cells. So it's not just one pathway of RNA silencing. There's several different pathways, and they're involved in regulating genes in different ways. And also, as I'll talk to you in a moment, they can act at the DNA and the chromatin level. So this then is this is the evidence that this is an RNA, a defense mechanism against viruses. So basically, when you look at plants that are infected with viruses, you see virus-specific small RNAs accumulating. If you make a plant that is actually knocked out for part of the silencing pathway, it is hypersusceptible to virus disease. And I think the real, the clincher is when you look at the genes that viruses have, you find that they have genes that are suppressors of this RNA silencing process. So you're probably all aware that if you want to look at evolution in action, the best way to do is always look at disease and disease agents, because there's this ongoing trench warfare, if you like, or arms race between hosts and pathogens. So as soon as a host evolves a resistance strategy against a pathogen, the pathogen will involve a counter-defense strategy. And then we can also see counter-defense strategies and all that sort of stuff. And sometimes you can see it happening in experimental systems. So if you want to look at evolution, looking at host-parasite interactions is very nice. And what we found in viruses is they have proteins which are able in various ways to suppress the RNA silencing process. So this is exactly what you would predict. If it's a defense mechanism, the virus evolves a counter-defense measure. This is one of these suppressor proteins, and its structure has been determined not by us, by other people. But it basically, it's quite nice. It forms a clamp around the small RNA molecules, and it stops them. Once they've been diced, this clamp comes in, and it stops them being recruited by slicer. Anyway, so this is it's a virus defense mechanism, and it does a whole bunch of other things in cells. I needed to show you this about virus defense, because what I want to do is now show you an experiment. And I've been running a lab for 30 years, and this is the coolest experiment that we have ever done. And we still haven't fully come to grips with the implications of this experiment. So the experiment is based on the idea that the RNA silencing mechanism is part of a virus defense system. So what we thought we'd do is fiddle around with our viruses, and the colors are all a bit funny here, but they would look a bit funny anyway. But we did an experiment with plants into which we had introduced a gene for the jellyfish green fluorescent protein. So this leaf is supposed to be green, and somehow it's come out as red. What we did was we took a virus and we introduced into the virus part of the coding sequence of the jellyfish green fluorescent protein gene, and we inoculated it to these plants. What was happening now was that instead of being green, as you can see on that slide there, this leaf was now red. So this was telling us that the green fluorescent protein gene was switched off. And that was what we expected. We knew that this is a virus defense mechanism, so we've got the coding sequence here. It's part of the virus. When the virus goes into the cell, you get small RNA molecules being made. RNA silencing is targeted against anything that's in the virus, including this coding sequence. So this gene gets switched off. So that's fine. What we wanted to do, this was the interesting bit, was we said, okay, let's not only look at the coding sequence, let's look at the bits of the gene that are adjacent to the coding sequence in the DNA. So these are bits of the gene that we thought would not exist as RNA, but we just wanted to know what would happen. So this was a sort of fiddling around experiment, if you like. What we found was that in this experiment, the leaves were red, as they were here. So that the gene was switched off. The jellyfish green fluorescent protein gene was switched off. So that's green, remember, and these are red. So we've done two types of experiment here where putting a bit of a gene into a virus causes the gene to be switched off. So what this told us was that somehow, and we've got other data, through the RNA silencing pathway, you could actually do something that affected the way that the gene, the DNA, was turned into RNA. But then what was really amazing was when we looked in the next generation, and it's such a shame that you can't see it. I'm going to turn my laptop round so that you can maybe see it, so that you know that I'm not making this up. But the progeny of these plants here were green. So you can see, green. So the gene had come back on here. And this is what we expected, because the virus is not transmitted from one generation to the next. But in this plant, the progeny of this plant here, the seedlings stayed red. There they are, they're red. So what this meant was that we had infected a plant with a virus, we'd plugged into the RNA silencing pathway, we'd silenced the gene, and what had happened was that the gene had gone off, and then it stayed off. It stayed off in the next generation, and the next, and the next, and we took it through about 5 generations before we stopped doing it. So the gene just stays off. The sequence of that gene hasn't changed. You know, the sequence of the DNA is completely as it was before. But somehow that gene has got a heritable characteristic of being silenced. So that's what happens when you fiddle around with these things. Anyway, so it's only water, it'll evaporate. Oh no!
Do you need anything?
No, I think we're okay, thanks. So what's going on here? This is all a bit, it's all a bit technical, but what is happening is that, remember, this is the slicer, and it's bound to the RNA. And it's somehow binding to DNA and causing actually a chemical modification of the DNA. It's not changing the A's, C's, G's, and T's, but it's adding a methyl group onto that DNA. And there's a whole pathway that explains all of this, and it involves small RNA, and I'm not going to go through it in detail, just to say that it's just a different flavor of the sort of RNA silencing mechanism that I showed you in the cartoon. So we've then got RNA silencing, and it can do something that we call epigenetic. It's an epigenetic control mechanism. It introduces heritable variation into DNA. And I think this is immensely interesting. So it was thought originally that epigenetics was all about genome defense. So we know that the elements of the DNA in plants and animals that has a propensity to move around, to jump around. And when DNA jumps around, it can jump into the middle of a gene and it would disrupt the gene, it would mess the gene up. So it's obviously not in the interest of a plant or an animal that it has these jumping genes hopping around all of the time. So it was thought that one of the reasons of these sorts of epigenetic mechanism, these heritable silencing mechanisms that I have just illustrated to you, were that they're somehow affecting the movement of these jumping genes. And this is just a picture of jumping genes in action on a maize cob. So this is the work of Barbara McClintock, who first described these. And when you lose the color on the maize cobs, that's an indicator that one of these jumping genes has gone into one of the genes in the maize plant. But it's not that simple. And one of the reasons why we know it's not that simple is because if we take a plant that can now no longer do this RNA silencing business, what we expected was that all of the transposons, the jumping genes, would go wild, but they don't. And actually the plant looks pretty much okay. So if this process that we're looking at, that we've just spent 10 years in sort of working out these complex cartoon-like mechanisms that I showed you and glossed over in the previous slide, if this whole process is doing anything, surely if you knock it out, then the organism should fall apart, or you should be able to see something. Well, what we've come to appreciate is that we were actually looking in the wrong way at what this epigenetic mechanism actually does, what this silencing mechanism does. And the reason, the little clue that we got that took us in that direction was actually when we looked at where in the life cycle of the plant are these small RNAs being most abundantly made. And we found that they were most abundantly made in the seed. And then we said, well, you know, where do they come from? And what we found was that in the seed, they're only derived from the maternal genome, and not from the paternal genome, which was a bit weird because once the seed has started going, then most of the other genes are expressed from both mum and dad. But these were only made from mum. So this then made us start thinking about all sorts of things like parental conflict and stuff like that. And I can talk about that a bit more in the discussion if you like. But the general idea that we were thinking about was that maybe a role of these small RNAs is not so much in controlling transposons or in regulating growth and development, although they do that to some extent. But maybe they have a different role in controlling the interaction between genomes. So remember, when you're a plant out there in the wild, then you're going to be hybridizing with every bit of pollen that comes your way. And those bits of pollen, some of them will bring in good genes, some of them will bring in bad genes, some of them will bring in new bits of transposon. And so you might want to be fairly discriminating against, you know, the sorts of pollen that you're prepared to hybridize with. And so if you've got a suppressive agent from mum that can control, you know, which dad makes babies, then that might be a good thing. And this is where we're going in this work at the moment. So we've derived this hypothesis that we're exploring, which is that the small RNAs, many of which are derived from transposons, allow the genome in an organism to define what is self and what is non-self. They allow it to discriminate when they have the good fortune or the misfortune to be hybridized with a different type of genome. We've done some experiments to explore that. So this is the sort of thought experiment that we were thinking about here. So just imagine you've got a hybrid. You've got a plant, or indeed an animal, but we think about plants mostly. We've got plant A and plant B. And the genomes of these plants are a bit different. You know, they're different varieties of plant. They might even be different species. So plant A will produce small RNAs that plant B doesn't make. And plant B will have targets of small RNAs that plant A doesn't make. So when these two mate, when they're hybridized, what happens is that the small RNA from this parent finds a target in the RNA of the other parent. They come together and they do silencing. So what you can propose, what you can hypothesize, is that when you make a hybrid plant, you get silencing that happens in the hybrid that doesn't happen in either of the parents. So we've looked into this using tomatoes. And there are various reasons for using tomatoes. They're very good experimental systems, as well as being nice to eat. And basically, our hypothesis is correct. And again, these are all sort of technical things, but I hope you can see, here's a graph, and in one hybrid line here, there's something going on, which is not going on in the parents, or indeed most of the other hybrids that we looked at. This silencing is that this graph is indicative of silencing of a particular gene, a gene that happens to be important for making the vascular system of the plant. It happens to be important in the defense of the plant against pathogens. So this is an indicator that when you make hybrids, you've got stuff happening that to do with silencing, as we'd predicted, that is resulting in genes being silenced that are not silenced in either of the parents. This is a whole phenomenology known as transgressive segregation. So it is, you see it in people, you see it in plants. Actually, so plant breeding or even evolution is actually dependent on this. So in plant breeding, what happens is you take two inferior parents and you cross them together, and out of that you get a superior offspring. Some of you may say that that sort of thing happens in human beings as well, but I wouldn't care to comment on that. But you cross together, so you've got two parents being crossed together. You've got things happening in the hybrids that aren't happening in the parents. So this contravenes sort of Mendelian genetics, if you like. It doesn't happen all of the time. It's the exception But nevertheless, it could be quite an important process in terms of evolutionary mechanisms. And so, I mean, this is where we're going at the moment in terms of our interest in RNA silencing. It happens, I'm told, when you get to be more than 50-something, and I'm now more than 50-something, you develop an interest in evolution. So you lot have got some way to go. But anyway, this is where we've got to. And it's what we're doing right now is we're trying to explore the extent to which these epigenetic and these RNA silencing mechanisms can explain transgressive phenotypes. So why it is that hybrids aren't always the average of the parents. So I've roamed around a little bit. I've given you a bit of stuff about viruses and defense. I've given you a bit of stuff about transgenic plants. We've delved into biotechnology and even biomedicine just briefly. And then we've finished up with hybrids and what happens when you make hybrid organisms. So I think with that, I should probably close. I'll just acknowledge, so Attila and Charles and Andrew have done a lot of the work that I talked about. Andrew Hamilton did the key stuff about the small RNA silencing, and Shiva has done the stuff about the hybrid plants. So anyway, thanks for your attention. Yep.
Could you perhaps clarify for me what you were saying earlier in the talk about the infective process
Right.
and getting resistance to that, in the light of what you were saying in the latter part of the talk about the interaction between genomes?
Yep.
And what role the small RNAs might have in that. Because I wasn't clear the first time around how the cellular mechanisms could differentiate between the RNA which had come in and the endogenous RNA. Is it the small RNAs that are helping to do that? Differentiation?
No, you're absolutely spot on. So in the cartoon that I showed you, we had the RNA polymerase coming on and latching on to the transgene RNA, or it would also do it to virus RNA, and turning it into a double-stranded thing. So why is it that that molecule, or those molecules, enter the pathway, whereas other cellular RNAs don't, or at least most of them don't? So what we think is that, we hand-wave a little bit. We talk about aberrant RNA. So if you've got a normal RNA, that is an RNA, a cellular RNA, what we know is that it actually has a structure where one end is bound to the other end. And it's got a sort of closed structure. And that closed structure stops the RNA polymerase from entering it and turning it into double-stranded RNA. When you make a transgene, we're not that clever. So sometimes the RNA molecules that are made from a transgene don't have this closed RNA. So sometimes the polymerases drop off a bit early, or they don't start at exactly the right place. So you've got a fraction of the molecules that don't have this closed structure. And then those can be then used by the RNA polymerase, and so it enters a sort of cycle. So that's what happens with transgenes. You know, you've got what we talk about as an aberrant RNA. With viruses, the same thing is actually happening as well. So we think that the viral RNAs are somehow recognized as being aberrant. So then you might say, well, why does the virus do that? Surely it would want to protect itself from this silencing mechanism. It's going to the trouble of making proteins that suppress RNA silencing. So why doesn't it just make decent RNA molecules instead of aberrant RNA molecules, and then it wouldn't enter the pathway? The reason for this, we think, is, remember, a virus is absolutely dependent on its host. The last thing a virus wants to do is to wipe out its host. So there's an interesting dynamic here, which is, although the RNA silencing is a defense mechanism against the virus is actually using that defense mechanism to downregulate itself. I mean, you can't teleologize these things, but this is how it's turned out. That is, the silencing process is, yes, it's a defense mechanism used by the plant to stop the virus, but it's also a process that the virus can use to stop itself going wild, if you see what I mean.
So, is the idea then that once this process has stopped going,
Yep.
then it's sort of upregulated enough that it overcomes the endogenous genes? Because presumably they're still protected
Yep.
in the way that you said. So at some point that's going to break down if it's turning off.
Yeah, no, well, I've spared you some of the sort of mechanistic detail, but there's a positive feed-forward element to this whole process. So once it's got going, it actually amplifies itself.
Any other questions?
Yep.
One of the things about dark matter is that there is a lot of it.
Yeah.
It seems to be a lot of this small RNA inside the cells. What about between the cells and out in the environment? I would assume it's getting broken down eventually, but there should certainly be a lot of leaking out.
Well, that actually, I could have given you a whole other talk on that, and we've done some work on this in my lab over the last several years. So one of the features of this whole process that we cottoned onto fairly early is that actually these little small RNA molecules can move between cells, and actually they can spread throughout the plant. And so they're part of a signaling process that plants have. And they use this in defense. So obviously if you've got a virus which is moving through the plant, if as part of a defense system you've got an antiviral molecule, a small RNA that can move with or ahead of the virus, then that is a good thing. And we believe that, well, we've got evidence that that sort of thing is going on. So yes, they do. These RNA molecules, they do move. They are intercellular, certainly in plants. Now, there's some indication as well in worms that they are intercellular. And recently, this is more controversial, but even in mammals and people, there's evidence that there may be some of these molecules actually circulating in the bloodstream. And I know there's a lot of interest in this, so I think the physiological function of the circulating RNA molecules is yet to be understood, but their presence can certainly be used as a diagnostic of disease in certain instances.
So just following on from that, would that mean if they can move between cells, then one doesn't have the old argument about the germline being kept apart from the somatic cells. So is that a possible mechanism for the Lamarckian inheritance?
Well, I mean, so firstly, I'm a plant biologist. In plants, we don't have a separate germline in the way that exists in animals, so we can get around that one. There is, however, some evidence for environmentally induced transgenerational effects in people. Studies in Switzerland, in Sweden in the late 19th, early 20th century, indicating that either smoking or poor diet, particularly in adolescence, can have effects that are manifest 2 generations further on. Studies of Dutch people during the war when there was extreme starvation, again indicating effects in later generations. So one starts wondering whether in mammals there is some process that allows communication with the germline. And, well, there are the molecular pieces in place, but whether or not they've yet been put in place, whether or not we can say that this is actually what is going on, I don't think we can at present.
Any other questions? No? All right. Well, we just give a quick reminder that next week we're going to be hosting Professor Lloyd Peck. We'll be talking about life in the frozen state. So please join us again next week, and also we'll be having elections after the fall next week. So once again, please give me a warm welcome. Thank you.
Thank you.